Welcome to the TOEFL Listening section. In this section, you will listen to an academic lecture and then answer questions about what you heard. You may take notes while you listen. You will have time to review your notes before answering the questions. Listen to part of a lecture in a university class. You will hear the lecture only once, so listen carefully. After the lecture, you will answer some questions about what you heard. Some questions ask about the main ideas and supporting details. Other questions ask you to make inferences or connections between ideas. Answer each question based on what is stated or implied in the lecture.
Good morning. Today we will focus on the components of the immune system, specifically white blood cells and antibody production. I want you to understand not only the names of the cells involved, but also how they interact to recognize and eliminate pathogens. We can group white blood cells into two functional arms. First, there are innate cells that respond quickly. These include neutrophils, which are the most abundant circulating leukocytes and are excellent at phagocytosis. Macrophages patrol tissues, ingest microbes, and present antigen. Dendritic cells are crucial sentinels that capture antigen and migrate to lymph nodes to activate adaptive responses. Natural killer cells are part of the innate arm as well. They recognize stressed or infected cells and induce cell death without prior sensitization. All of these innate cells set the stage for adaptive responses by controlling infection early and by producing signals that shape the later response. Adaptive immunity is mediated mainly by lymphocytes. B lymphocytes produce antibodies after differentiation into plasma cells. T lymphocytes include helper T cells that provide help to B cells, and cytotoxic T cells that kill infected cells. The collaboration between helper T cells and B cells is central to effective antibody production. Let us follow a simple pathway for antibody production. A B cell with a surface receptor that recognizes an antigen binds that antigen. If a helper T cell recognizes a linked peptide presented by a professional antigen-presenting cell, the helper T cell provides activating signals. The B cell then undergoes clonal expansion, differentiates into plasma cells that secrete antibody, and some progeny become memory B cells. Initially, antibody responses are dominated by IgM. With T cell help, B cells can undergo class switching to produce different isotypes, such as IgG, IgA, or IgE. Class switching changes the antibody constant region, altering effector functions without changing antigen specificity. Affinity maturation occurs in germinal centers through somatic hypermutation. Mutations in the variable region that increase affinity are selected, resulting in higher affinity IgG antibodies during the course of an immune response. This process is essential for producing effective neutralizing antibodies against evolving pathogens. Antibodies perform several key functions. They can neutralize pathogens by blocking entry into cells. They can opsonize microbes, coating them to enhance uptake by phagocytes. Antibodies also activate the complement cascade, which promotes lysis and clearance. These mechanisms are distinct but complementary. Consider a practical example. In influenza infection, innate cells limit early viral replication. Dendritic cells present viral peptides to helper T cells. B cells initially make IgM, but after class switching and affinity maturation, high-affinity IgG and mucosal IgA are produced. IgA helps at mucosal surfaces, while IgG circulates and neutralizes virus systemically. Memory B cells formed after the primary response are long-lived. Memory cells respond more rapidly upon re-encounter, producing large amounts of high-affinity IgG. This is the principle behind vaccination. where exposure to antigen in a controlled way builds memory and permits a quick, robust antibody response when the real pathogen is encountered. There are clinical applications that illustrate the importance of these components. Measurement of neutrophil and lymphocyte counts informs us about innate versus adaptive activity. Therapies such as monoclonal antibodies mimic natural antibodies to neutralize toxins or block receptors. Understanding which white blood cells and which antibody isotypes are involved guides treatment choices. To summarize, the immune system uses a diverse set of white blood cells and the adaptive production of antibodies to detect and eliminate pathogens. Innate cells act first and inform adaptive cells. B cells, with T cell help, produce antibodies that neutralize, opsonize, and activate complement. Memory B cells and class-switched high-affinity antibodies provide long-lasting protection. When you study this material, pay attention to the sequence from antigen recognition to clonal expansion to differentiation into plasma cells and memory cells, and to the specific functions of antibody isotypes.
Listen again to part of the lecture, then answer the question.
Memory B cells formed after the primary response are long-lived. Memory cells respond more rapidly upon re-encounter, producing large amounts of high-affinity IgG.
This concludes the listening section. For more TOEFL practice tests and study materials, visit Spinta.org.